Good afternoon, everyone. Today we will discuss our new topic, that is bacteriology of butter. So, bacteriology of butter differs in many respects from that of milk because the composition of butter is such that it is less favorable for the growth of microorganisms because it contains more fat. Therefore, it does not favor the growth and transfer of bacteria. Butter contains salt, and this salt is dissolved in the water and which is surrounded by a water droplet by the fat particles, and salt inhibits the bacterial growth. Butter is usually preserved at low temperature which does not favor the growth of bacteria, but some molds may grow in the butter. Molds is the vegetative growth of the fungus. Then we will discuss about the bacteriology of meat. Meat is an important source of nutrition for the mankind. Meat must be wholesome and safe for the consumption. Meat is spoiled due to the action of microorganisms, and these microorganisms may get entrance through the following sources. So let's see the sources. The first source is the animal itself. The animal which is diseased is a major source of microorganism. Such diseases are transmitted to human being by consumption of contaminated meat. Therefore, there are the following types of meat-borne diseases. Meat-borne diseases of chemical or toxicological origin. Second one, animal diseases transmitted through meat to man. So every disease of zoonotic importance should be regarded as potentially meat-borne diseases. What is meant by zoonotically important diseases? Means the diseases which can be transferred from animals to the human beings. That diseases we call as the zoonotic diseases. Because in course of handling and slaughtering of animal, then dressing for meat purpose, numerous opportunities arise for the transferring of infective agent from animals to man. So these may be the sources of infection to the mankind. The infection enters through digestive tract, abraded skin, and respiratory tract. The important meat-borne diseases are here. In first column the name of the diseases are given, and in the second column their causal organism or causative agents are given. So anthrax is a meat-borne disease. The causative agent is the Bacillus anthracis. Tuberculosis, tubercle bacilli. Brucellosis, Brucella abortus. Salmonellosis, Salmonella species. Typhoid, Salmonella typhosa. And botulinum is the Clostridium botulinum. These are the meat-borne diseases, means zoonotic diseases, and in second column their causative organisms have shown here. Okay. Then environmental contamination of meat. How the environment contaminate the meat we'll discuss. Certain strains of staphylococcus in meat and meat products cause gastrointestinal disturbances without any change in appearance and taste. The troubles are diarrhea and vomiting. Sources of microorganism is also due to unhygienic condition of persons who handling the meat, unsanitary condition of butcher's shop, restaurants, etc. To avoid bacterial contamination through atmosphere, keep the meat in glass or plastic boxes, and provision of fly screens for doors and windows in butcher's shop. It helps in reducing the chances of staphylococcal contamination. Refrigeration is one of the impressive methods of inhibiting the bacterial growth. Then we will discuss about the bacteriology of fish. Bacteria and their toxins in a fish and fishery products make them poisonous. They may affect central nervous system and create gastrointestinal and skin disorders. The more important types of bacterial food poisoning are caused by the Salmonella, Staphylococci, Clostridium botulinum, Escherichia coli, and Streptococcus facialis. Then we will discuss about the bacteriology of egg. The fresh laid egg, if perfectly clean and sound, are normally free of bacteria, both with respect to the shell and the contents. So long as the shells are kept dry and clean, bacterial contamination should not be there either in a fresh laid, held, or stored egg. The mucinous nature of cuticle of shell tend to prevent bacteria penetrating the cell. Also, the shell membrane has bactericidal property to some extent. Bactericidal means it's preventing the growth of bacteria or killing the bacteria. It is the bactericidal property. The bactericidal property of albumin of fresh laid egg prevents access of bacteria to the yolk. Alkalinity of stored egg is unfavorable to growth of bacteria. So egg stained with fecal material, soil and moistened shell results in bacterial decomposition. So especially if shell becomes moistened, cracked in transport, is quite susceptible to bacterial invasion. Then we will discuss the bacterial defects in the egg. First one is the black rot. The defects in egg are large movable air cells filled with gas, greenish brown, watery void, that is albumin, and black yolk. Broken egg gives foul smell and appear rubbery. Bacteria responsible for black rot are alkaligens, Escherichia, and Proteus group. These are found in soil and fecal material. Then second one is the green rot or sour egg. Albumin undergoes certain degree of liquefaction, that is stringy. It appears green in color. The yolk is covered by pink and white spots, look like partially cooked yolk. Green rots are caused by genus Pseudomonas, a soil microorganism. In early stage, the infected eggs are very difficult to detect. Then red rot. Bacteria of Pseudomonas group gives rise to distinct reddish tinge in yolk. This can be detected by candling. So candling is a technique by which we can detect these rots, or we can detect any defect in the egg yolk or egg albumin. We can detect the defects of the internal part of the egg by candling. The albumin is usually liquefied and has grey cast containing reddish colored patches. So this is all about this topic.